All right, so I'm going to try and cover pretty broad area in 10 minutes. So this is where we're moving. Again, there's a major lack of evidence in almost every single one of these potential mechanisms, and hopefully in the coming years we have some answers. Okay. Each one of our cells has approximately a 10 micron nucleus. And inside that 10 micron nucleus is 2 meters of DNA. So that's the equivalent of taking the Empire State Building, coiling it up, and trying to squish that into a ping pong ball. And later when we eat, because of epigenetics, our neurons don't produce gastric acid because the genes for gastric acid are coiled up in a way that they're inaccessible to RNA polymerase and they're not expressed. Our brain doesn't liquefy when we eat a meal. I'm going to talk mostly about DNA methylation. That's one of the chemical post-translational modifications that changes and alters that coiling of the DNA, allows certain genes to be expressed and other genes to not be expressed. In general, when a gene is hypermethylated, it is less accessible, hidden inside the coil, and not expressed. Okay, so in 2013, there was first evidence that infection induces epigenetic changes. Ben Youngblood published that 2 years after successful aviremia, people living with HIV had hypomethylation of PD-1 and hypermethylation of IL-2. There was another paper, my slides are moving. There was another paper in schistosomiasis that again, after successful treatment for schistosomiasis, there were persistent epigenetic changes that lasted at least as long as 6 months. There was a study in hepatitis C, 82 weeks after successful therapy, after sustained viral response, there were persistent DNA methylation changes. And then finally, there was a TB study published around 4 years ago. CD4s, CD8s, CD14s, and NKs all had persistent DNA methylation changes despite antimicrobial success that lasted 6 months after the end of successful therapy, 12 months from diagnosis. So what? So what's the clinical relevance of these changes? Now again, the HIV fields had already shown that HIV, the epigenetic changes induced by HIV led to 4.7 to 5.2 years of premature epigenetic aging. So we asked, did this premature epigenetic aging happen in TB? And yes, there was 12 years of premature epigenetic aging in our small cohort with very small numbers. Now, luckily, I was working with a very talented bioinformatician, Carly Bobak, who then used an RNA-based clock and found that this premature aging also was validated with 14 years of premature aging using a second RNA clock. This aging correlated with oxidative stress and senescence. Now, senescence is the failure of immune cells to die. And when there's an increase in immunosenescence, that's associated, not in TB studies, with increased cardiovascular disease. So this is pretty low-hanging fruit. If you have these immune cells that are failing to undergo the regular BCL2-induced cell death, they're releasing pro-inflammatory proteins, IL-1 beta, IL-6, TNF, CXCL9, that probably are increasing atherosclerosis. Now, the next mechanism I want to talk about is metabolism and epigenetics. The cancer field has really demonstrated quite nice. So we've known since the 1920s that cancer and infections induce this Warburg effect, this shift from oxidative phosphorylation to glycolysis. Now, when this persists for too long or is too strong, Leo showed the beautiful data on glutathione depletion. Now, glutathione is going to replenish our NADH to NAD ratios. When NAD is too increased, there is a response that sirtuins are activated, which leads to epigenetic changes. Similarly, when the TCA cycle is wound up too much, succinate and itaconate are elevated, and those inhibit a major set of epigenetic enzymes called the alpha-ketoglutarate-dependent dioxygenases like DNMT and TET, and result in these very broad epigenetic changes. The third mechanism is related, is the increase of cellular ROS, which activates an enzyme called PAC1, expressed by DUSP2, which again induces these pretty significant epigenetic changes. We tested this in an LPS and a TB-induced immune tolerance model, and what we found was that metformin, everolimus, and N-acetylcysteine all prevented the TB and LPS-induced hypermethylation. And in fact, if you gave itaconate or succinate, it would induce the same infection-induced DNA hypermethylation. So in addition to dysregulated metabolism leading to these epigenetic changes, it can also lead to persistent inflammation via lipid peroxidation. So again, we've known for over 100 years infections increase glycolysis. When that increased glycolysis doesn't have the glutathione system to reduce these rogue electrons, we get lipid peroxidation, and the ER, the mitochondria, and the cell membranes get ruined. When that happens, CD36 positive macrophages will take up those oxidized lipids, resulting in foamy macrophages leading to atherosclerosis. Similar to the very nice data that Leopold showed and was published elsewhere, this is preliminary data from Eswatini. You can see that the CRP levels are not normalizing at the end of successful therapy, and more than half of the microbiologically cured individuals have CRP levels still greater than 3. You can also see that despite successful therapy, lipid peroxidation, as measured by H&E oxidized LDL, remains elevated at the end of successful therapy. In the bottom left corner, the Trolox assay is of reducing capacity, very similar, again, to what Leo previously showed on the glutathione system. And then the last thing we checked is because the major nutritional source of reducing agents in your bodies is from carotenoids, we measured mass spec carotenoid levels. And again, what was previously demonstrated in a couple of studies is that low carotenoids would increase your risk from TB progression. So we see here that the TB patients do have lower carotenoid levels than their household contact controls, but you can also see that they're not normalizing at the end of therapy. So we were investigating why they might not have normalized, and what we did is we looked at the DNA methylation status of those who did normalize to those who did not normalize. And what you can see here is a heat map of different pathways, and the people that normalized their carotenoid levels had DNA hypermethylation, that epigenetic silencing mark. So this is suggesting that the epigenetic silencing is allowing the immune system to calm down and for carotenoids not to be consumed in that lipid peroxidation. But we still have a lot to test that. Here's the contaminated slide. I'm sorry. What this slide would have shown if the heat map showed up is we looked at 3 groups, resolving inflammation in Eswatini, and then improved FEV1 from a cohort from Dr. Wallace, improved FVC from that same cohort, and improving resolution, resolving inflammation by CRP or improving lung function was associated with DNA hypermethylation. In contrast, when we looked ex vivo at MTB killing capacity, that was all hypomethylated landscape. Sadly, if this data holds up, it suggests that there's a trade-off. By inducing a hypermethylation, we can resolve inflammation at the expense of being immune responsive, whether it's for recurrent TB or a secondary cancer. So a lot more work to be done, but it's a little concerning. Two more things I want to briefly talk about. One is clonal hematopoiesis, and if you read articles on clonal hematopoiesis, it says that infections will increase it. Okay, so we're told every cell in our body has the same genetic makeup, but as we get older, as you're over 50, around 10% of people have single nucleotide polymorphisms in their stem cells. The genes that it most commonly occurs are listed right there. 3 of them are epigenetic enzymes, and one of them is a major regulator of the immune system. People with these have 2 major problems, an increase in leukemia and MDS, and an increase in cardiovascular disease. Again, no TB studies here. This is something that has to be taken out of review articles as a possibility, and we need hard evidence. The last one, especially based off of yesterday's really interesting conversations, is here's 4 studies, not TB studies, that took individuals who had no known respiratory disease, no known respiratory symptoms. The top 2 were in Germany, then one in Molise, Italy, and then the last one in South Africa. And what all of them showed is that lacking respiratory symptoms or disease, if you had decreased FEV1 or FVC, there was increased all-cause mortality and cardiovascular disease, maybe suggesting that lung damage, even asymptomatic lung damage, is leading to inflammation and driving cardiovascular disease. So we're lacking a lot of evidence in this field, and it's something I'm really hoping we can change as a community over the coming years. And these are some of the things that I think are driving that persistent inflammation. It's really great to hear the field embracing endotype concepts, and we probably have to think about what's driving different endotypes. When air pollution is driving hyperinflammation, when the downstream metabolites of isoniazid are driving inflammation, when there are epigenetic scars that are driving inflammation. So thank you, and questions at the end.